The Chernobyl wolf. 6 generations have lived inside the exclusion zone since 1986. Scientists collaring these wolves do not measure acute exposure. They measure chronic saturation. These animals never leave the radiation field. They are born in it. They hunt in it. They sleep in the same contaminated soil where firefighters once buried their melted dosimeters. Every breath, every drink of water, every mouse they eat carries a whisper of cesium-137. Natural selection is visible in their DNA. The fastest evolving regions of the wolf genome are now cancer immune pathways. Their immune systems run in a state that, in humans, only appears during radiation therapy. Activated, scanning. Aggressive. The radiation did not adapt them. It filtered them. Over 6 generations, wolves without specific genetic variants died. What remains is a population being bred by radiation itself for cancer resistance. These wolves are not surviving despite the radiation. They are surviving because of what the radiation removed from their gene pool. The weak died, the lucky died. The ones left carry something new. The Princeton lab studying this is not a wildlife lab. It is a cancer lab. They ask whether 40 years of natural selection in Chernobyl can teach human oncology something about immune surveillance that $30 billion of research has missed. The wolves are already teaching them. Their T cells recognize and destroy precancerous cells with a speed that human T cells cannot match. The difference is not in one gene, it is in a network, but the wolf still carries the same basic mammalian vulnerability. To find true radiation tolerance, you have to leave mammals entirely and stop measuring in millirem. The cockroach pop culture has a single answer to nuclear apocalypse: cockroaches. The reputation is wrong. Cockroaches are the weakest animal on this list. They are famous for surviving the bomb, but that fame is based on a misunderstanding of what survival means. The German cockroach tolerates 900 to 1050 gray. A human dies at 5. That 200 to 1 ratio sounds like a superpower, until you learn the secret. Cockroaches barely divide their cells. Insects only undergo cell division before molting. Radiation kills by shredding DNA during replication. No replication, no shredding. The cockroach is not tough. It is just slow. Its cells stand still while human cells sprint into the gunfire. If you force a cockroach to molt, if you trick its cells into dividing, its radiation resistance collapses to human levels. The armor was never armor. It was a calendar. Even at survivable doses, 10 gray sterilizes them. The nuclear apocalypse cockroach kingdom does not happen. They cannot reproduce afterward. The ones you see after a blast are the last generation. They will not produce more. Some ant species tolerate 2,250 gray, more than double the cockroach's ceiling. Ants share the same slow division strategy, but they have added something else. Something about their social structure, their shared immune memory, allows them to recover reproductive function after doses that sterilize cockroaches permanently. The lesson here is negative. Radiation tolerance is not about armor. It is about timing. The less often a cell divides, the less often radiation finds it vulnerable. But waiting to divide is passive defense. Some insects have moved beyond passivity, into a radical chemical strategy that no mammal possesses. The cockroach teaches us what does not work. The next animal teaches us what might. The fruit fly. The fruit fly is the most studied organism in genetics. Every biology classroom uses it. For 60 years, radiation biologists have used it to discover something the cockroach hides: that survival is not the same as recovery. Drosophila has a radiation tolerance of approximately 640 gray, lower than the cockroach, but mechanistically more interesting. The fruit fly possesses a family of proteins called scavenger receptors. These detect and neutralize oxidative damage before it propagates. More importantly, Drosophila males recover fertility after sterilizing doses through a process called germline stem cell repopulation. Damaged stem cells self-destruct. Healthy neighbors replace them. The fly does not just survive, it replaces what it loses. This stem cell replacement mechanism is absent in mammals. Human ovaries and testicles cannot replace their own radiation-damaged stem cells. The fruit fly can. The difference is evolutionary lineage, not complexity. The fly keeps a reserve population of dormant stem cells that mammals do not maintain. Labs actively study Drosophila germline stem cells. They ask whether mammalian stem cells could be induced to perform the same self-replacement after chemotherapy or radiation exposure. If a human testicle could learn the fruit fly's trick, male cancer patients would not lose fertility. The mechanism is not magic. It is molecular signaling. The fly releases a protein called unpaired that tells healthy stem cells to divide and replace the dead. Human cells have a similar protein, IL-6, but in humans it does not trigger replacement. It triggers inflammation. The difference is in the receptor, not the signal. The fruit fly still needs its cells to survive radiation directly. One microscopic animal found a different path. Not replacement, but protection at the chromosome itself. The tardigrade 8 legs, half a millimeter long, and a protein that no other animal on Earth possesses. One that physically shields DNA from radiation before damage can begin. The tardigrade has become an internet legend, but the internet usually gets the science wrong. Here is the correct version. Radiation does not shred DNA directly. It splits water molecules inside cells into hydroxyl radicals. Those radicals do the shredding. The tardigrade produces a protein called DSUP, damage suppressor. It binds directly to nucleosomes, wrapping the genome in a molecular bodyguard. When hydroxyl radicals arrive, DSUP intercepts them at the chromosome surface. The radicals still form. They just never reach the DNA. This is not repair, it is pre-armor. Human cells with the tardigrade DSUP gene inserted suffer half as much DNA damage under radiation as normal human cells. The protein works across species. A tardigrade adaptation already functions inside human tissue. Cancer radiotherapy kills tumors while damaging healthy tissue. If DSUP can be delivered specifically to healthy cells, leaving cancer cells unprotected, it would transform treatment. Several biotech companies now hold patents on DSUP delivery vehicles, viral shells that carry the tardigrade gene into human bone marrow. The tardigrade is not a curiosity. It is a patent, but DSUP prevents damage. What if you could instead survive the damage, not by blocking it, but by protecting the machines that do the repair? The bedelloid rotifer. This microscopic animal has survived 1,000 gray and remained reproductively active 2 weeks later. At that dose, even tardigrades are sterilized. Here is what makes no sense. Radiation shreds rotifer DNA just as badly as it shreds anyone else's. You can measure the double-strand breaks. They are identical in number and severity to those in sensitive species. But something else survives that does not survive elsewhere. The repair machinery itself. Bedelloid rotifers resist radiation-induced protein carbonylation, the destruction of their own repair enzymes. Every other organism loses its ability to fix DNA because the fixers get broken. The rotifer protects its repair shop. At 1,000 gray, its DNA is confetti. But the machines that reassemble confetti are still running. The workers are in an indestructible bunker. Their genomes contain thousands of genes stolen from bacteria, fungi, and plants through horizontal transfer. Some of their radiation resistance may literally be bacterial DNA plugged into an animal body. They are not a single species. They are a fusion, a living archive of stolen genetic code. The engineering problem here is radically different from tardigrades. Not shielding the building, but armoring the repair crew. That distinction is something most materials science has not even begun to explore. But the rotifer still requires water to live. Remove water completely. Dry an animal to a husk. Something stranger happens to radiation chemistry. The midge larva. This African midge larva can be dried to less than 3% of its body weight in water. In that state, it is biologically dead. No metabolism, no movement, no respiration. And in that state, radiation cannot touch it. Hydroxyl radicals require water. No water, no radicals. When the larva dries out, its cells replace water with trehalose, a sugar that vitrifies into glass around every organelle, every protein, every strand of DNA. The larva becomes a crystal with genetics inside. Radiation passes through, but without water to split, there is no secondary destruction. The midge never evolved radiation resistance. It evolved drought resistance. Radiation resistance came free, a side effect of being able to turn into a biological statue. Larvae have been rehydrated after 17 years in desiccation. They resumed movement within hours. The same cryptobiotic state that pauses death also pauses radiation chemistry entirely. Dry transport of living cells, vaccines, tissues, and blood products currently requires freezing. Freezing destroys most biological samples. Trehalose vitrification is already being adapted for human cell preservation. Several labs have successfully dried human blood platelets in trehalose and rehydrated them weeks later with no loss of function. But the midge still makes a trade. It must be dry to be resistant. One insect found a different solution, one that works wet, works dry, and works while carrying only half the normal number of chromosomes. The parasitic wasp, male Habrobracon, carry one copy of each chromosome. Human cells carry two. The backup copy is supposed to be essential for radiation survival, a spare blueprint when the first is shredded. By that logic, these males should be fragile. They are not. Habrobracon males survive 1,800 gray, nearly double the cockroach's ceiling, 360 times the human lethal dose. The paradox is explained by repair speed. Diploid cells wait to find a matching chromosome for homologous recombination, a slow, precise process. Haploid cells cannot wait. There is no matching chromosome, so they use faster, single-strand repair pathways that do not require comparison. The absence of a backup forces a faster primary response. The wasp turns vulnerability into processing speed. Habrobracon was sent to space in the 1960s, specifically to study radiation damage. It was one of the first animals weaponized against cosmic rays as a research tool. NASA used it before they used most mammals. Understanding how haploid cells accelerate repair without a template could change space radiation protection. Backup systems take mass. Speed takes only biochemistry. The wasp still has an upper limit around 2,000 gray. One organism on Earth has no meaningful upper limit, and it was found in a can of meat. Deinococcus radiodurans. In 1956, a can of ground meat was sterilized with gamma radiation, doses high enough to kill every living thing ever discovered. The meat spoiled anyway. Something was still alive inside. The scientists who found it thought their equipment was broken. They recalibrated. They ran the experiment again. The same result. Something was eating the radiation. Deinococcus radiodurans tolerates 5,000 gray during active growth, 1,000 times the human lethal dose. At that dose, its genome shatters into hundreds of fragments. The bacterium does three things no other organism does simultaneously. First, it keeps a high manganese to iron ratio. Manganese scavenges the radicals that iron would amplify. Second, it organizes DNA into tightly packed toroids that physically hold fragments near each other. Third, it uses a repair mechanism called extended synthesis-dependent strand annealing, reading overlapping fragments like a jigsaw puzzle and stitching them back together error-free within 24 hours. At 5,000 gray, it emerges genetically unchanged. Not just alive, identical. Hiroshima survivors received 3 to 6 gray. This bacterium survives 5,000 gray. The comparison is not a ratio. It is a category error. It was discovered because it refused to die in food designed to contain no life. Deinococcus enzymes are already used in PCR testing. They tolerate the heat and chemical disruption that destroy other biological reagents. The bacterium's manganese chemistry is being studied for radioprotective drugs. Its genome assembly mechanism is informing synthetic biology. How do you store information so that even after catastrophic fragmentation, it can be rebuilt perfectly? The bacterium is the end of the numbers game. There is nothing on Earth, nothing discovered so far, that survives more ionizing radiation while remaining reproductively intact.